In this presentation, we are going to examine transcription and synthesis of messenger RNA. That's the first step in synthesis of proteins. Transcription is the process of copying a segment of DNA into RNA. An RNA transcript may play various roles in a living cell. It could be a protein coding gene, or it may participate in protein synthesis or some other activities, including processing of other RNA molecules. For a given gene, only one strand of the DNA serves as template for transcription. And here is a short sequence of double-stranded DNA. The top strand, shown here in red, is the coding strand. The bottom strand, shown in blue, is the template strand. It's also called minus strand or the sense strand. This strand is a template for synthesis or copying of DNA into messenger RNA. Enzyme RNA polymerase synthesizes messenger RNA. And that's actually what happens in prokaryotes. In eukaryotes, direct copy is heterogeneous nuclear RNA or hnRNA. So that's direct copy. Copy occurs in 5' to 3' direction, complementary to the template strand. So the opposite DNA strand in red is called the coding strand, non-template strand, the plus strand, or the antisense strand. The easiest way to find the corresponding RNA sequence, which is shown here in green, is to read the coding strand directly in 5' to 3' direction and substitute thymine of DNA for uracil in RNA. In a nucleus, DNA is in highly compact form in chromosomes where it is tightly wound around histones. Alterations in histones and the rearrangement of nucleosomes may help expose DNA sequences for transcription. Acetylation of lysine disrupts ionic interactions between its ammonium ion and phosphate groups of DNA. And that helps unwind part of DNA from histone and make it accessible for translation. Only short segments of DNA are unwound and transcribed at a time. Typically, hybrid DNA-RNA double helix has only 8 to 9 base pairs. Complete unwinding of DNA from histones would be energetically unfavorable, and newly synthesized RNA would become tangled with DNA. In previous presentations on tertiary structure of DNA, you have learned that double strand of DNA is wound around histones and that lysine and arginine residues serve as templates for double strand of DNA to wound around protein histone. That occurs through salt bridge interactions, through ionic interactions. And so salt bridge holds together a side chain of, in this case, lysine and a phosphate group of sugar phosphate, deoxyribose phosphate backbone. When it is acetylated, when side chain of lysine is acetylated, then those salt bridges or intermolecular interactions are disrupted, they are no longer possible, and that segment of DNA unwinds from the histone. RNA polymerase is the enzyme that catalyzes transcription of DNA to RNA. Bacteria have only one type of RNA polymerase. Eukaryotes have 3 types. RNA polymerase 1 transcribes ribosomal RNA genes. RNA polymerase type 2 transcribes protein coding genes, that means synthesis of messenger RNA, and RNA polymerase 3 synthesizes transport RNA and other small RNA molecules. Transcription begins at a DNA sequence that is known as a promoter. A gene to be transcribed must be recognized by a regulatory factor. In prokaryotes, an example of regulatory factor is sigma factor. In eukaryotes, a set of general transcription factors interact with DNA at the promoter to form a complex that recruits RNA polymerase and may further alter chromatin structure. At the active site of RNA polymerase II, the two DNA strands separate to form 12 to 14 nucleotide transcription bubble. When the correct nucleotide forms hydrogen bonds with a template base, a change in conformation of enzyme brings the catalytic residues in a position and forms the bond. The bond is formed by a nucleophilic attack of the incoming 3' hydroxyl of the nucleotide on the 5' phosphate of the polynucleotide. That means that RNA molecule grows in 5' to 3' direction. Within the 12 to 14 nucleotide transcription bubble, hybrid DNA-RNA double helix has only 8 to 9 base pairs. Transcription and translation differ in eukaryotes and prokaryotes. In prokaryotes, a messenger RNA transcript is typically translated immediately without any further processing. In eukaryotes, transcription results in synthesis of heterogeneous nuclear RNA, hnRNA, which is the actual precursor of messenger RNA. Primary RNA transcripts of eukaryotes, so hnRNA, then undergoes further processing. First, it includes addition of a 5' cap structure, which protects 5' end from 5' exonuclease. At 3' end, a sequence AAU-AAA is the signal for a protein complex to cleave the transcript. So that causes termination of the transcription and attaches a protective polyadenyl tail of about 200 adenyl residues. DNA of eukaryotes has or have intervening sequences of DNA, so-called introns. And introns separate coding fragments of DNA, which are called exons. So simultaneously with processing 3' and 5' ends of messenger RNA that are just described, splicing carried out by RNA protein complex called spliceosome, composed of 5 small nuclear RNAs and hundreds of proteins, joins exons and eliminates introns. Basically, introns are cut out, exons are joined together. So once that happens and exons are joined together to form contiguous stretch of DNA, that's messenger RNA. Messenger RNA then leaves the nucleus for the next step for translation. Translation occurs in the cytoplasm. And we'll cover translation in the next presentation. Now, sometimes genes are silenced or not expressed. Silencing means that gene is not expressed after transcription. Such post-transcriptional gene silencing involves small interfering RNAs, siRNAs, which disrupt gene expression by forming double-stranded RNA, which are then degraded. Ribosomal and transport RNA transcripts also undergo further processing, for example, introduction of unusual nucleotides in transport RNA. They are processed by nucleases and enzymes that modify particular bases. Small nucleolar RNA, snRNA, catalyzes chemical modifications of transport RNA, ribosomal RNA, and DNA. So this completes our presentation on transcription, and in the next presentation we are going to examine briefly translation.